Okay, so let's talk about how you process the images that you took last week. So last week you performed the walk-in protocol where you had about 10 files in an empty vial. So your vial looked cleaner than mine. You didn't have the stuff at the bottom, and you also don't have the stopper. So the first thing you need to do is have ImageJ. So if you search ImageJ on any web browser, it will bring you to this screen. You can download it. This is traditionally what has been used, but now they allow you to run it in the browser. So whichever one is best for you, and we will be using this every week when we analyze our pictures. Okay, so it's a very simple program, and it really was created to help scientists analyze different images and things that we capture in the lab. If you go back to the Drosophila protocol, and I'll also attach it with this video, there's a packet on what we're doing here, and it tells you the process. So you will go to File, Open, and you will open this document wherever you have it stored on your computer. Then you're going to go to the line tool, and you're going to draw a line. You're going to trace a line that's on your grid paper. My grid paper is smaller than yours, so 2 squares on mine is a centimeter, but yours is 1. Okay, so make sure you are able to trace one line on it. Then you're going to go to Analyze, Set Scale. Okay, you're going to say that this distance is 1 centimeter. Make sure that global is unchecked. Okay, so your known distance is 1, your unit of length is a centimeter. Okay, so now that's telling you when we measure this, that's allowing all of us to be able to compare our images. Because remember, some of you are comparing males to females. Well, if you have males in your vial, you need to know that whoever has females in their vial is doing this in the exact same way as you, so that we can analyze it and compare them. Alright, so the next thing you're going to do is choose this multi-point selection, and you're going to click on that, and what you're going to do is set each point. So the first one is going to be the bottom. For you, it's going to be here. Okay, for me it's here, because that's the bottom where my flies are starting from. So remember, yours is going to be the very bottom of your vials, the starting point for your flies, and you're going to click it. Then after you click it, you're going to click the center for each fly. So 1, 2, 3. Okay, I have 4. You should have hopefully around 10. So however many flies are in your vial, you're going to click on them. And so I have 5 clicks here, but remember that first click is the bottom of your vial. The next thing you're going to do is go to Analyze and Measure. Now this is calculating the distances for you. So save this. So Files, Save As, wherever you save your files. And make sure that the name of the file matches your picture or some way that you know. So this can be Results, Vial 1, 2, or Picture A. Okay, because remember you have 3. So Vial 1A, B, and C would all be the same vial the 3 times that we took it. Then you're going to say save it, and you're going to exit out of it because you don't want to save these results, because we're going to do this again. And then you can also exit out of your picture. Okay, you don't want to save it. Now you're going to open each of these over and over and over again. So remember that you should have 3 images for each one so that we can take the average of them. But you should have however many flies you have. So for some of you, it may be 1 vial. For some of you, it could be 3 or 2. Okay. So then next, you're going to open up Excel. And that's where you're going to analyze the data that you captured in ImageJ. So I'm going to open the one I had. Nope. So open it. So after you open it, remember that this is your starting point. So now what you're going to do to get your distance, so make a new, and your distance is equal to this. Let's see, the best way to do it would be to type it in. So type in 20.98 minus each cell, and then you can carry down. So my flies walked 6, 5, 6, 2. Okay, and so then you will repeat this for each vial, and then we can get an average to see how these flies are walking, or we can keep it as a scatter. So for now, all you would need to turn in is this for each one. Okay, this is my vial for 1A, but if I have B, I can do something like, okay, 1A here, 1B, and then 1C, and then you can make another sheet, and you can do, this will be results for vial 2. Okay, and then you can keep all of those together. And so you will turn in this Excel spreadsheet too, so that I can keep track of your data, and we'll have all this data together when it's time to analyze. Okay, so if you have any questions, so make sure you go read over this, look at this video, see if it comes up with any questions, try it for yourselves. Everyone in your group should be able to do this. Any questions, comments, concerns, contact myself or TA immediately so that you're on the right track. Okay. Good luck.